with cells and organelles. The first thing I'll do, sort of define organelle, and we're going to actually also discuss the types of cells that exist on the planet, of which there are 2 kinds. But first, I want to talk about these cells, or organelles, that we find within cells. So an organelle is a specialized structure devoted to a particular function within cells. So let's write a subcellular, so within cells, a subcellular structure with a specialized function. So we're going to be talking about 6 or 7 specialized function. We're going to talk about 6 or so organelles today. Specialized function. So we find these organelles devoted to particular functions, and we find them within our cells. And let this guy in. If you look at the word organelle, it sounds like organ, right? It's like a mini organ, I like to think of it. So like our body organs are pretty much have the same definition of having a specialized function. Our heart pumps blood around the body. Our pancreas has a specialized function of secreting digestive enzymes and insulin to regulate blood sugar. Our stomach has a specialized function of digesting food. Our liver has a lot of specialized functions, but you know, they're all very unique, right? And specific. And that's exactly what we find with our organelles. But they're just mini organs within cells, microscopic, right? Very small. So we're going to look at a few. And the first one we're going to look at is the nucleus, the cell's control center. Now, I'm sure maybe some of you are thinking, didn't we already discuss the nucleus this semester in the last unit? Who remembers the nucleus from last unit? Different nucleus. We talked about the nucleus of an atom, and the nucleus of an atom is that dense core in the center of our atoms that contain the protons and neutrons. Yeah, they happen to name them the same, annoyingly, but the nucleus of a cell is very, very different than that. So let's define it. The nucleus of a cell looks like that. And there's our cell. So the nucleus, the primary function of the nucleus, it's really its main only function is to store DNA, protect it, house it. We call it housing the genetic information of our cells. And it just acts as this protective vault, because of course our DNA, as we'll see later on in the semester, is extremely just important when it comes to cell function, to body function, growth and development, etc. And so the nucleus is an organelle just devoted to containing it. So that's our first one, nucleus. And there's much more to be said about DNA and the nucleus and how DNA itself when it comes to genetics, but that's exactly it. This unit we don't talk about genetics yet. So we're going to leave it there and keep it as a sort of foundation to just have in your head of, okay, the nucleus of cells contains DNA, contains genetic information in the form of DNA, this polymer of nucleotides, the nucleic acid we talked about last unit. And later on, we'll discuss more at length sort of what role the nucleus plays in what I said about cell function, body function, these types of things. Okay. So the next organelle we're going to talk about, a very critical organelle here. It's called the endoplasmic reticulum. It's a long, fancy name. And if you notice, the endoplasmic reticulum is just right outside the nucleus, kind of borders it. So I'm actually, oh, and if you take a quick look, we have the rough endoplasmic reticulum and the smooth endoplasmic reticulum. I'm just going to go to the board, going to write some things about this fascinating organelle. Okay, how's the lighting? Is that a little better? So the endoplasmic reticulum long, crazy name, so we actually call it the ER a lot. So ER. So the ER is made of stacked membranes, actually. Kind of like a flattened series of membranous sacs just stacked on top of each other. And there are two types of endoplasmic reticulum. Two types. There's the rough ER and the smooth ER. So there's rough ER, and I think I'll have room, smooth ER. So I find it best to kind of tackle two in one here. We're going to talk about these two types of ER, but there's another organelle that I guess, I read in the textbook, they don't technically call it an organelle, but you could think of it as one. That's specific to the rough ER called ribosomes. And we're going to define that here too, just kind of do kill two birds with one stone kind of thing. But the primary role here of the rough ER, it's devoted to protein synthesis, specifically proteins that are going to leave the cell, be exported from the cell. So we're actually going to first write, well, it's called the rough ER for a very specific reason. We're going to say it's attached to ribosomes. or rather ribosomes are attached to it. It gives it the rough appearance. So I'll just show you on the slide. Here's your rough ER, and you can see our ribosomes are attached to it. And I have a kind of funny analogy here, or comparison, but you can think of it as like a poppy seed bagel, right? Kind of looks like that. And then the Smoothie R is like a plain bagel. Anyways, I was hungry one day and looking at it, and I was like, that looks like a bagel. So the rough ER, you have these ribosomes attached to the stack of membranes. And then let's go ahead and define ribosomes. Ribosomes are the structure or organelle responsible. There we go. So ribosomes, let me just do parenthesis. They're the organelle where protein synthesis occurs. So I'm going to write organelle responsible for protein synthesis. So that's what ribosomes do. So whenever proteins are being made, proteins are being manufactured, amino acids being linked together, it happens at the ribosomes. And the rough ER is coded in these ribosomes. So the primary function of the rough ER is to make proteins. And sort of in this outward flow where the proteins made in the rough ER are specifically made to be exported from the cell. So we can even add that. So I'm going to actually erase this. Attached to ribosomes where proteins are made for export from the cell. Now, you may be wondering what kinds of proteins that might be for export from the cell. Well, let's name a few. There's a few fascinating, important examples of proteins that are constantly being made and are secreted from cells, such as the pancreas, pancreatic cells that are actually connected to our small intestine by a tube, pancreatic duct. There are cells lining the duct in the pancreas that actually secrete digestive enzymes, which are proteins, right? So digestive enzymes are a great example. I should add examples, sorry. So, examples, right? I meant to write that, sorry about that. Examples, digestive enzymes. Another great example are white blood cells called B cells. Synthesize antibodies all day long against infectious agents like viruses and bacteria. And those antibodies are proteins that are exported from the B cells. So we could write antibodies. Again, this is all going to be made at our rough ER. And you could add insulin too if you want. Insulin is also made in in the pancreas, different area of the pancreas. And insulin is released from cells into our blood supply and regulates blood sugar. B cells, you don't have to know what B cells are. They're just part of our immune system. So that's why I wrote antibodies. B cells will secrete antibodies, which are important in fighting pathogens. In fact, to become immune to COVID, your B cells, memory B cells, secrete antibodies against the virus, immune complex it. So whenever you're re-exposed, you have antibodies circulating around, and it'll immediately glob onto it and prevent it from invading our cells, actually. That's the concept behind herd immunity, generating that memory. It's also the concept behind vaccines and stuff. But I digress. That's the rough ER. It's the main function of the rough ER. Let's talk about the Smoothie R. What's the Smoothie R doing? The Smoothie R is doing a vastly different function. I'll just keep the, let's write, the second type is the Smoothie R. So, as its name implies, the Smoothie R does not contain ribosomes, right? So no ribosomes present. And interestingly enough, the Smoothie R has a few functions to it. So let's write responsible for first being lipid synthesis. So a couple examples would be steroid hormones like testosterone and estrogen. So we could write steroid hormones as an example. Also, membranes themselves are made of what we discussed, last time, phospholipids, right? Those are also going to be made by the Smoothie R. So, phospholipids for membranes. And another funny, interesting, different function of the Smoothie R is detoxification. So there are a lot of enzymes present in the Smoothie R that process alcohol, drugs, medication, things like that, and break them down. So actually your liver cells, specific zone of liver cells, have a lot of Smoothie R. They're packed with Smoothie R in order to do that. Okay. One more thing I'll mention before we move on. Cells will have, right, rough ER with ribosomes attached, Smoothie R, but they also, you might be wondering, right, if the rough ER ribosomes are devoted to protein synthesis for proteins that are leaving the cell, what about proteins that stay in that cell? Because obviously that's important, right? So they're in a cell, you know, you have your nucleus and then you have your ER with you, and the rough ER is dotted, you got some Smoothie R, whatever it is. But you're also going to have free-floating, unattached ribosomes in the cytoplasm. And those are devoted to protein synthesis for proteins that stay in the cell. So unattached ribosomes. You can write assemble, if I can fit it here, assemble proteins that stay in the cell. How am I going to fit that? Assemble proteins that stay in the cell. So that would be some other examples we've discussed. We talked about structural proteins. So our skin cells are packed with keratin. So keratin would be made on these unattached ribosomes. Metabolic enzymes, of which we're going to talk about at length this semester. I mean, this unit, we're going to talk about metabolism in the cells. But I just want you to know that these unattached ribosomes make proteins that stay inside of the cell. They don't leave. The rough ER is devoted to proteins that are flowing outward in the system. And that's a great transition for our next organelle. Because, you know, proteins aren't just synthesized at the rough ER and they just bail. They've got to be processed, they got to be modified, and they got to be packaged up. And there's an organelle devoted to that. And that's going to be our next organelle. It's called the Golgi apparatus. Any questions, though, so far before we move on to the next organelle in our list here? By the way, if just a cool thing I'm gonna get my connect. So I don't know how your guys' connection is. Like if I break up sometimes or it's kind of screwy. I had a student recommend to me yesterday at my other school to get an Ethernet cord. So I'm getting that on Amazon. So if that's happening, hoping that'll help. Just to have it directly plugged in versus Wi-Fi. So I'm hoping soon I'll have that going. Well, I'm going to order it today, so I'll probably get it Thursday. Anyways, no questions. All right, sweet. Then let's move on to our next organelle, the Golgi. So the Golgi sometimes has a bunch of names. Golgi complex, Golgi apparatus, Golgi body, any of them. Same thing. So we'll just call it the Golgi complex. I think that's what I have in my slide. Protein synthesis definition. Just creating proteins, manufacturing them, building them. Here we go. All right. So again, we have a series of flattened stacked membranes, just like our ER, but has a different function. As you can see, there's these vesicles that butt off, these little packages surrounded by membrane. And again, this Golgi is going to be devoted to this process of export and things leaving the cell, namely proteins. Although lipids synthesis, just fun fact if you're interested, in that vein, lipids synthesized by the Smoothie R can also exit the cell through the Golgi. Now, what is the Golgi doing exactly? It is modifying the products of the ER and packaging them for release from the cell. So sometimes they need to be tagged with a sugar. Sometimes they need additional help folding to become their final form and their final structure, these proteins. They're often tagged with a sugar because it's sort of like, I mean, you could actually, I love analogies and comparisons. So the Golgi is kind of like the FedEx of the cell. And it's kind of like, you know, if you want to send something somewhere, you got to put a address, the sending address, right? Delivering address. So FedEx knows where to ship it to. It's the same thing with proteins, funny enough. They often get tagged with a carbohydrate chain, a little carbohydrate, that will distinguish the protein and actually allow wherever it's going to recognize it and take it in or process it or whatever it is. But I just want you to know that we are modifying these products of the ER, packaging them. It's the FedEx of the cell. Yeah. Golgi's the scientists who discovered it. Scientists love to name things after themselves. Because we're show-offs. Well, not me, I haven't discovered anything. Devoted to? Just shipping, packaging up and shipping out products of the ER. Oh, I must have confused, sorry. The Golgi isn't tagged with anything. The products of the ER would get tagged by the Golgi. So the Golgi apparatus or complex has enzymes attached to its membrane or membranes so that when a product of the ER, so let me just show you here. So when a protein is synthesized in the ER here, the rough ER, it'll then go to the Golgi in one of these transport vesicles, that protein. The vesicle will then fuse with one of these membranes, and there are enzymes in the Golgi complex that will tag these proteins with a carbohydrate label. And when they're secreted after getting tagged, kind of like a stamp in FedEx and a shipping address, it actually it allows target cells that are going to take up this protein that it's being shipped out to recognize it and not treat it as foreign or anything. That's all. That's just how it's modifying things and packaging them up and shipping them. Okay. oftentimes, yes, Sophia, that's exactly right. Yeah. So oftentimes, there's tiny carbohydrate molecules attached to the surface of cells in order to identify it. And so actually our circulating white blood cells that are looking for foreign bacteria and viruses, they don't attack our cells. The reason is because there's a carbohydrate complex called MHC on all of our cells sticking out, and it says, hey, don't attack me, I'm part of Ryan, or I'm part of you, right? So the white blood cell that can digest it and destroy it and cause inflammation and kill cells says, oh, you're self, I'm not going to attack you. That's it. Don't worry too much about that. I just wanted to clarify what I was saying. This is the only level I want you to know. I just like this stuff so I can ramble on. Okay, let's move on to the next organelle, the lysosome. Okay. Now, the lysosomes are interesting. They have a very unique function. Yeah. Very different than what we've been talking about when it comes to proteins being made and exported from the cell, or or other molecules being made. The lysosome is devoted to digesting and breaking down molecules. So we're going to write that the lysosome's function is to digest / break down large molecules. And so lysosome, it's a membrane sac like this, or just a compartment surrounded by a membrane within cells that contains digestive enzymes. So let's just say these are our digestive enzymes. And the lysosomes play a few different important, fascinating roles inside cells. You could say this is the recycle bin of the cell. So it's digesting and breaking down larger molecules. When would it do this? There's a couple prominent examples. I think I have one on my slide here, one or two. Okay, so lysosomes are important let's start with damaged organelles. So if there's an organelle that's old or damaged, the lysosome will actually fuse with it and break it down, recycle it into its molecular components, and it can be reused to build proteins or membrane or whatever it is. So it'll do that. That process, it's actually, no, I won't get into it, but that's one role of the lysosomes, kind of that recycling, renewing function. Another role, this is actually very prominent, very important in unicellular, single-celled organisms, like amoebas. But essentially, if you're if you're one cell and you take in food, you got to break it down somehow. And single-celled organisms like amoebas will actually take in food from the outside, maybe algae or something, who knows, and inside that vesicle, it'll fuse with a lysosome and the digestive enzymes in the lysosome and break it down. Now, I do say amoebas because our lysosomes aren't as devoted to this sort of nutrient digestion process, only because we digest things in the stomach with acid, and we secrete enzymes into the small intestine. But for these unicellular organisms, their lysosomes are obviously very, very important. So this is important for our purposes, for sure. We don't want damaged organelles. It can, cause issues. But another really interesting, I think, amazing function of the lysosomes with us is going back to our immune system at a perfect time, the whole pandemic, in digesting pathogens, digesting viruses, digesting bacteria, breaking them apart, breaking them down. So we have certain white blood cells that will take in bacteria that we might breathe in or get in our food, into our GI tract. White blood cells will find them, and they'll do the same sort of thing. They'll engulf the pathogen, the bacterial cells, or the one at a time, bacterial cell or viral particle, and lysosomes will fuse with it and digest it, tear it apart, destroy it. And it's actually a process called phagocytosis. This is all things, if you continue biology, especially in microbiology and immunology, it's like lesson 1. You learn about phagocytosis. It's a hugely important immune process for host defense and being infection-free and killing off pathogenic microorganisms. It's awesome. So I just wanted to name a few relevant examples of lysosomes, but that's the main function there. Okay. So I might as well just write some examples of its uses. I don't know why I didn't before. Actually, I do. It took a long time at my other school, but might as well. So let's write breaking down old, worn out organelles. That's one use. Digestion of pathogens or disease-causing organisms. So let's write digestion of pathogens. And then for unicellular organisms, could write food digestion. For unicellular, meaning one cell organisms. And I'm actually going to say, like amoebas, because when we look at different cell types towards the end of the lecture today, we'll discuss how certain cell types don't have organelles. So amoebas happen to be made of the type of cell, or they are the cell type that has organelles, but there's a different cell type that doesn't have any organelles in it, but we'll discuss that. So just write unicellular organisms like amoebas that do have organelles like lysosomes rely on these lysosomes to digest food. Okay. Mitochondria. It's our next organelle. And who knows what the mitochondria do? What are they? What's the mitochondria's nickname? Yeah, it's the powerhouse of the cell. It's funny how that's like a common thing everyone knows, like as a yeah, nickname. It's the powerhouse of the cell, right? So that's right. Powerhouse of the cell. And what do we mean by that? The mitochondria is the location of ATP production. And I'm actually going to add, it's the location of ATP production through a process called cellular respiration, which we're going to have 2 amazing lectures on soon enough. But mitochondria are a powerhouse. And remember, ATP is this energy currency of the cell. It's that nucleic acid molecule that's packed with energy. It's like a battery. And so for any process that requires energy, which we're actually going to discuss next lecture, we need ATP. It's like our battery. Just like my phone has a battery, it requires energy to power on. There are certain things in our cells, certain mechanisms, certain processes that require ATP molecules because they require an energy input. So the mitochondria are key, the key main organelle for massive production of ATP. And it looks like this. It's like a bean-shaped organelle, and fascinatingly enough, it has 2 membranes. It has an outer membrane, and it has an inner membrane. That's kind of folded and squiggly around like that. And the inner membrane that is folded and loopy a little bit, the folds, we call them cristae. Just a fun fact. So these folds are called cristae. Alrighty. So we have now covered every organelle that exists in animal cells. So before we move on, I just want to do a cool, fun little review of identifying organelles. And we're going to look at, and note that I said animal cells. We've covered all the organelles, but we haven't covered all the organelles, unless I have cytoskeleton in here. Do I? Oh, I do. Okay, so let's cover this really quick. The cytoskeleton, not technically an organelle, but nonetheless, very important structural component of cells. And here are some of the functions of the cytoskeleton. Essentially, cytoskeleton is made of these different kinds of proteins that there's a ton of them throughout cells. The whole cell is filled with these microfilaments and intermediate filaments and microfibrils, these types of proteins, microtubules, microfilaments. And here are some of the functions of the cytoskeleton. The shape of the cell is maintained. Things moving around the cells are moved along these microtubules. They're important in cell division, movement of the cell itself. They're important, and they anchor the organelles in place. So I did want to mention the cytoskeleton. It's not to be ignored. It's still an important integral component here. And I actually have a really cool picture. So when you stain cells with fluorescent dyes, you can see just how abundant these fibrous proteins are that run throughout the cell to give it its shape. So this looks like a typical skin cell. And in the center there, that's the nucleus. But you can see all these fibers, protein, cytoskeletal elements running through it. Okay, here's my fun activity. Oh, and okay, anyway, sorry. So, all right, let's have some fun. So I'm gonna ask you guys what organelle is being pointed at. So let's start with H. What is H? Yeah, there's our mitochondria, double membrane, bean shape, cool. How about A? What's A? Yeah, that's the nucleus, absolutely. How about D? Yeah, ribosomes, nice. How about F? Tricky. Yeah, Golgi. Yep. The F is the Golgi complex. How about C? ER. What kind of ER is C? The rough ER. Absolutely. Making B the Smoothie R. Poppy seed versus plain bagel. How about G? Kind of tricky because, but the hint is I just talked about it. Yeah, cytoskeleton. Yeah. I forgot I had that in my drawing, so that's why I talk about the cytoskeleton. Definitely important. So anyways, how about eye? What's the fluid called in our cells that surrounds our organelles? It is. It's the cytoplasm. Yeah, cytoplasm. Okay, and then the last one I think we haven't talked of was E. What's E? Yep, E are the lysosomes. Just those sacks with the digestive enzymes inside. Awesome. D is ribosomes. Yeah, D was ribosomes. Yep. F was the Golgi complex. Okay. Nice. B, let me go back to it. B is the Smoothie R. Okay. Now you'll notice I wrote eukaryote in parentheses, right? Yeah, you can print this out. It should be on Canvas. Like I mentioned, right, we've talked about all the organelles we'd find in an animal cell, like our cells, but we haven't mentioned organelles that we'd find in a plant cell. There's actually 3 additional structures, organelles that we'd find in addition to these. Bubbles not colored in. Oh, those must be vesicles budding off. Just little membrane compartments that would hold proteins or lipids synthesized. You don't have to know that. It's just that, you know, because that's how the rough ER and Smoothie R would transport things throughout. They are contained in these membrane vesicles, we call them. Okay. So plant cells, actually, interestingly enough, plant cells have all of these organelles that we just talked about, but they actually have 3 additional organelles. Well, 2. One of them is a structure that's additional on top of all of these. So let's talk about those. And since they're plants, I'll use green. So our plant cells have 3 additional structures. Three additional structures. But I do also want to mention and emphasize again, because sometimes this is a misconception of sorts. All the organelles in animal cells are present in plant cells as well. So let's write, have all the same organelles as an animal cell. And then, of course, they have 3 additional structures. The first additional organelle is called a chloroplast. And the chloroplast is devoted to photosynthesis. So this is the organelle where photosynthesis takes place. I think we will have time to discuss cell types today. The second structure that plants have that animal cells do not is the cell wall. Cell wall. And we talked a little about the cell wall in the last unit. You guys remember what it's made of? It's made of what? Talked about it when we talked about carbohydrates. Yeah, cellulose, exactly. So cellulose was that polysaccharide, that structural polysaccharide. So plant cells have a cell membrane surrounding the cell, of course, but then they also have this additional polysaccharide structural cell wall surrounding the cell membrane, giving it that rigid, firm, supportive structure. Polysaccharide. And it's why whenever you have salad or whatever, there's a crunch. You're crunching through the cellulose. Versus animal cells were all flabby. Just membranes for us. Okay, the last neat structure in plant cells is called a vacuole, a central vacuole, specifically a large central vacuole. Large central vacuole. And the vacuole just stores water. Stores water. And it gives plant cells this enlarged shape to the cells. Think of it kind of as a water balloon that's full versus just have a little water in it and it's all, or less water, it's kind of not as enlarged. And it kind of, without the vacuole, it's going to wilt. So plants that have all this water stored in their vacuoles of the cells are going to be what's the word? Supported. I don't know, but yeah. Oh, really? Oh boy. Can you guys hear me now? Okay, well, luckily I'm getting this Ethernet set up soon, so yeah, that happened at my other school too. So I'm getting this Ethernet and I'm hoping that'll help this situation. Okay, but you can hear me now. So basically what I said was this large central vacuole stores excess plant cells, giving it the enlarged shape of these cells, giving it a fullness, additional support versus like a wilted, crumbling. It froze again? Mother. No. Okay. Man, that's super annoying. Okay. Can you guys hear me? No freezing? I'm good? Okay. Well, to repeat for the third time, the central vacuole stores excess water, gives the cells this full shape, rigid shape. It gives it the enlarged, yeah. Anyways, 408. So the next thing I want to talk about, oh, well, let's take a look actually at, oh my goodness, wait. Okay. Zoom freaked out on me for a second there. Okay, you guys can see my screen here. Here's just some pictures of, here's the chloroplast where photosynthesis occurs. Okay, sweet. Photosynthesis occurs in our chloroplast here. This is a cell wall, picture of cellulose in the plant cells. And then here's our central vacuole, just a giant excess water storage organelle. Okay, so what I'm thinking of, well, let's see, 409, let's see. So I think this will be a good, 5 minutes. Let's see how much I want to write. Yeah, it's probably too much. So this is a good place to end, even though we're ending early, which I never do. But well, I'll just leave you with this. There are two kinds of cells. There are prokaryotic cells and eukaryotic cells. And just as so we're going to talk about the differences next time at the beginning of class. But I just want to tell you, just as a sending off note, I suppose. Well, first, if you'll notice, go back to sharing here, prokaryotic cells are very small compared to our eukaryotic cell here, much larger. But a key difference between our prokaryotic eukaryotic cells is that eukaryotic cells, our larger cells, have organelles. Specifically, they have membrane-bound organelles. And I mention that because sometimes ribosomes are considered organelles, but they're actually not membrane-bound. And you'll actually find ribosomes in prokaryotic cells too. But I think next time I'm gonna talk about or emphasize that point again. I think it's a good point to end. Plus, we have the lab mini lecture to do. But yeah, because I need more time to talk about the differences. Okay. So any questions? And then we can kind of take a 10-minute. Ribosomes, they are organelles, but I think the textbook might say they're not. It doesn't matter. They are specialized structures within cells. I think sometimes they don't call them organelles because they're not surrounded by a membrane, a phospholipid bilayer membrane. But I've seen them called organelles. I've seen them not. I'm just going to emphasize when we talk about cell differences between prokaryotes and eukaryotes, organisms made of eukaryotic cells versus organisms made of prokaryotic cell. They're all single-celled. That's why I said that in that way. The eukaryotic cells have membrane-bound organelles. Prokaryotic cells do not. Both of them have ribosomes. Ribosomes are key to life on Earth, life in any capacity. Ribosomes are key. So any cell you find will have ribosomes. But these membrane-bound organelles, like chloroplasts, like mitochondria, like lysosomes, you're not going to find them in prokaryotic cells. But this is, again, just kind of food, you know, keep in the back of your mind for next time. We're going to really dive into the differences. Any other questions before we'll take a 10-minute break before the lab mini lecture. I'm glad you can hear me now. Make that order tonight on Amazon. Okay, so let's come back at 4:23-ish for the lab mini lecture. Sound good? And yeah, 10 minutes. Just go to the bathroom, get some food, play some tunage. Okay.
Take my hand and come with me because you look so fine that I really want to make you mine. You look so fine that I really want to make you mine. 4, 5, 6, come on and get your kicks. And you don't need the money when you look like that.